All right, good afternoon. Today in class we sort of ended on a mathematical cliffhanger. We ended up with this really abstract graph called a hill plot. Here it is right here. And this is the last thing that we ended with, and then I promised that we would make this connect to health and to people's lives. Just want to make sure this is working. It's been kind of a day for technology not working, but it looks like we are good. All right, so let's move away from the land of graphs where we want to turn life into a straight line. It's very powerful. It's why you have to learn it. But also getting away from the straight lines, how can we take this information and use it to heal people? And I'm going to give you 5 medical applications of hemoglobin biochemistry. Some of these will get a little specific. Sometimes I'm going to show you like a recent finding that's going to be a paper. And of course, the specifics of the recent finding are not important. But if I'm showing you at a slide in lecture, there is a single slide level concept or idea or image that I want you to take away from it that's really important. And so I want you to think about that. If I'm taking the time in lecture to show you a slide, there's some way I want you to be able to use it. And I want you to be able to use this information as a doctor in the field, maybe even as a doctor practicing global medicine in Africa, which is where sickle-cell anemia is more of a problem than it is here. So let's talk about the 5 medical applications. We'll kind of move up from the simplest one, which is acidosis. If you are familiar with some of the kinds of diabetes, if you have too much of a certain kind of molecule produced in the body, it will make the pH become more acidic. Not a lot more acidic, it's a highly buffered solution, so just a couple of fractions of a point. But that's enough to shift hemoglobin binding, and in fact that's one of the early experiments that they did. So one of the first things you do when you figure out how to get a binding curve of hemoglobin is that you want to add acid to it, add base to it, see what happens. And there was a scientist named Bohr. He was actually the father of the famous Bohr, Niels Bohr. He was a biochemist, so who knows what my kids will do someday. But this is called the Bohr Effect for the original father of Niels Bohr. And I forget his name. I just know his last name was Bohr. So notice that as we move away from neutrality, as we add acid, the pH goes down. The binding curve also goes down. That means for a particular PO2 value, you will have less theta, less YO2, less fractional occupancy of your hemoglobin sample. And the question is why? Well, you can actually look at the parts of the structure we've already focused on to ask the question why. If you remember this set of salt bridges, alternating charges, it holds the T conformation tight, T for tight. This is what gives it its name. So you remember that histidine. You remember that there's a couple of things, and you know what the pKa's of these things are about. And the one weird thing about histidine is that its pKa in solution as a free molecule is pKa 6. And those can shift by a unit or more in the complex environment of a protein. Which means if we're looking for a group that could accept a proton or donate a proton, it could shift its pKa, it's around 7. That would be the kind of group that could account for the Bohr effect. And if we think about it, the direction of the movement also accounts for the Bohr effect. If you add more protons, that histidine is actually going to actually get more positive charges. Adding more protons means that that histidine will be more likely, it might be like 6 times out of 10, it's going to be up in this configuration. You add more protons around, and maybe 9 times out of 10 it's going to be up in this configuration, which means the hemoglobin as a whole will be pushed more towards the T state, which means tight at this interface and less able to bind oxygen. A higher KD, a worse KD for binding oxygen. So one of the salt bridges we've already looked at very well could explain the Bohr effect just by itself. This is one of many, shifts hemoglobin toward T away from oxygen binding. This is just the one that we've shown you before, so I'm showing you it now. If you look at it more closely, you can see right here that the added proton literally forms a bridge. forms a plus between the two minuses of the N and the O, the N in histidine and the O in the aspartate. And it's bridging the alternating residues, and it holds these four residues sort of closer together. There are other ones. If you want to know what the other ones are, you can actually find out how people have investigated it. There's a whole paper about this by a scientist I admire named Martin Karplus, who actually is a computational scientist, but who looked at this and figured out, okay, it's not just beta 146. You see, beta 146 is the biggest factor, but there's also alpha 89, alpha 50, beta 116, and they add up so that when you're moving away from pH 7, you're actually moving to more protons being released upon oxygenation. In other words, as oxygen binds, you will actually release more protons, meaning the oxygen binds, it breaks that salt bridge, it will release a proton. But this gets complicated, and this is the trick for this whole chapter. You want to be able to look at the thing that you're told is changing and say what else changes. So in this case, if you add protons and you decrease oxygenation, then if you if you add oxygen, then you will do the opposite. So adding protons causes oxygen to be released. Adding oxygen causes protons to be released. And you see how this is all an equilibrium type reasoning. And the question is always, what am I adding, and how will the system respond to my adding it? And there's even more parts to this, and they involve protons as well. Because it turns out that carbon dioxide, and now I've been saying CO2 accidentally all along, now I mean to say it. This is CO2 can bind to the exposed N-terminus. The part that we were just talking about was at the C-terminus of the globin domain, right? Well, it turns out that the N-terminus is situated so that the carbon in the CO2 can actually bind to the amide, the amine, at the very N-terminus of things. When it does that, it forms a covalent bond and it kicks off a proton. That means, but think about that a little bit. If you have a lot of CO2, you're in actively respiring tissue, and you're in a place where you want to bind that CO2 and you want to release more oxygen. So what this does is the CO2, if you're deep in the tissues and you've got the hemoglobin still holding on to some of its oxygen, CO2 will bind the N-terminus, release protons. Those protons will protonate the salt bridge. They'll cause it to be more in T state, and that will cause its affinity for oxygen to lower, which means that oxygen will be released. Look at this. If you add, oxygen and CO2 are sort of opposites in respiration, right? You breathe in oxygen, you breathe out CO2. Here's a case where if you have, it's sort of the opposite. If there's a lot of CO2 around, it means it needs more oxygen because it's doing more respiration. A lot of CO2 around means protons are released, means oxygen is also released. Very, very cool. So carbon dioxide and oxygen actually can compete for hemoglobin binding. If CO2 goes on, it changes the local pH and it causes oxygen to fall off. If oxygen goes on, that means the opposite will always happen. If oxygen goes on, that means it's going to cause carbon dioxide to fall off. I'm not sure if it's physiologically relevant, but you've got to, everything that you talk about that can go one way, it's the whole point of calling it reversible. It means if it can go one way, it can also go the other way. CO2 binding releases protons, forms additional salt bridges, shifts hemoglobin to the T state, and therefore oxygen is released. If you go the other way, when there's a lot of oxygen around, CO2 will end up being kicked off the N-terminus, and hemoglobin will be forced more into the R state. The oxygen sort of forces the hemoglobin into the R state. That promotes the actual backwards changes from everything we're talking about right here. The bottom line, hemoglobin is an amazing molecule, and it's just the first amazing molecule that we're able to talk about in biochemistry, because it starts from a simple binding function, and it ends up being a sigmoidal curve that delivers oxygen most effectively. It helps it do two things well. Helps it bind oxygen in the lungs very well, and then helps it release oxygen in the tissues where the PO2 is lower. So pH change will also help it do these things. Carbon dioxide binding and release will do that through a local pH change. And so the thing about all this is I want to introduce you to Janet Iwasa, who is another scientist who I've been talking about her work for years because she has these really cool movies. So now that we've talked about hemoglobin, let's look at the movies. There's 3 movies. These are on Canvas, so I'm just going to talk you through them. And notice that she is showing you the R state, and she's showing you back and forth the R state in red, T state in blue. R state in red, T state in blue. And so then we're going to zoom out and we're going to see how the whole molecule changes. She visualizes, she's like the master pi-mole user, and is able to be an artist with these and showing. Remember, we talked about how the entire C-terminus moves toward itself. You can see there's that motion as it moves from the T state to the R state. When it binds oxygen, the C-termini close up close to each other. And all the other things change as well. Let's rotate around. Let's look at the dimer interface, and you can see the interface between alpha 1 and beta 2 is moving a lot more than the one between beta 2 and alpha 2. These really are dimers. Now I want to give you the chance to look at that over and over again and think about what it means. Here's another one of hers. Same idea. This shows you a T state, and it shows you the oxygen's trying to bind. One binds, and it shifts it to the R state. And you see immediately the other ones are shifted more red. One leaves and the other one leaves, and they don't cooperate yet until you get 2 that leave at the same time. And then they would do the same kind of cooperativity in reverse. Let's watch that again. All blue, 1, 1. It's not cooperative until you have 2 that come together, close together. And once you get 2 that come close together, then you quickly get 3 and 4. This is the nature of cooperativity right here. Because once you get the one binding, shifts the one residue to be more red. And when you have more red, you end up with the other domains being shifted to be even more red. So more red makes more red, and you have sort of this exponential growth of oxygen binding when you have that happening. Now, there's one more thing that we're going to talk about. It's BPG is what your book calls it. It also can be called DPG. And I want you to see this molecule, what it does. Here it is. It's between beta 1 and beta 2. It's right in the middle of the molecule. So if you look carefully at that, that's where the C-terminus wants to come together. So we have a molecule that wants to bind there. And when this molecule is bound, it binds better to the T state. When oxygen binds, it kicks this molecule off. Okay, just like that. Because it binds right in the middle, right where those C-termini come back together. You can look at that to your heart's content later. But that is one of the things about how hemoglobin works. It's just notice this shows you cooperativity. So I want you to have a feeling for what's really going on with the molecule. And Janet Iwasa's work helps you see that. Now what's interesting is you've probably seen some molecular visualization art that was done by David Goodsell earlier. And I've always liked his, I actually have his book. Oh, where is it? It's right here. My students actually one year saw I was using his stuff so much, and they actually got me the book. Here it is, The Machinery of Life. So he actually has a book, and if you see that, it's David S. Goodsell. And right here is actually signed. This is from the biochemistry class, and I believe it has David Goodsell's signature on the title page, right there. So I have the best regards of David Goodsell right there in his own book. I always liked him. He did this Molecule of the Month. He does this sort of molecular art kind of thing. But now he's retired, and I was like, who are they going to get to replace David Goodsell? And they turned to another molecular visualization artist, Janet Iwasa, who runs a whole lab that does molecular visualization. Here are some examples of David Goodsell's pictures, and now Janet Iwasa is continuing his work. I love it when I like somebody's work and then they retire and somebody else I like also fills their job. It's people doing what they're called to do. Find that thing for yourself, you know. You don't have to do everything, but you should find the something that you love to do that really is what you can do uniquely. For some people, it's combining art and biochemistry. Okay, so let's move through to some more medical applications of hemoglobin biochemistry. So let's talk about that molecule. That molecule in the middle is actually related to high altitude sickness. This molecule is 2,3-bisphosphoglycerate, BPG, or as other people call it, DPG. They just call it dye. And you actually will see this again in biochem too. It's one of the intermediates in glycolysis. It's where the cell gets the molecule from. For us, right now we don't care about where it gets it from. It's just that matters that it's around, and we can make more or less of it depending on the situation, which means that we can make more molecules to stick in the middle of hemoglobin to get in the way of oxygen binding. It turns out that they actually include this even in their erythromer, that blood substitute that we were talking about. They actually have that as part of the mix. It's a very important regulator. In fact, it's so important, if you try to purify out hemoglobin, you end up with a lot of BPG still stuck into it because it binds to it so tightly. Physiologically, you usually have a little BPG around, and the question is how much. So what does it do to our graph? Well, what it does, remember, it binds in the middle, and it promotes the T state, right? Because when the R state happens, it closes up its binding pocket. So that means if it promotes the T state, that means it's going to be associated with less oxygen binding, and it's going to actually shift the sigmoidal curve. It's going to shift that down. Look right here. Here's BPG at 8 millimolar, and BPG, that's in green. BPG at 5 millimolar, less BPG is the blue line. The blue line has been shifted up. Less of the molecule bound means more oxygen bound under the exact same conditions. But look at what that does. That shifts where the curve is exactly. And what you might be saying to yourself is, why am I making a molecule that makes hemoglobin work worse? Don't I want to bind as much oxygen as I can? But remember, hemoglobin does not do just one thing. It binds oxygen in the lungs. It has to let it go in the tissues. BPG actually helps it let go in the tissues. This is how the math works out exactly. And the other thing about BPG that you'll notice, the reason why you make more or less, it depends on the partial pressure of the oxygen around you. You will actually detect how much oxygen you're getting from the air, and if you are getting too little oxygen from the air, you will make more BPG. Well, how will that happen? For example, so remember that we talked about the partial pressure of oxygen is somewhere around 12 or 13. That's true for sea level. But as you move up on a mountain, for example, if you get on a plane and fly to Denver, you're suddenly at 4,500 meters up. The partial pressure of oxygen has dropped from 13 to 7. It's dropped almost in half. That means that when your lungs load up with oxygen, when your hemoglobin goes to the lungs, it's not at this point, it's at this point right here. And it's getting less oxygen in. So, what do we do? Do we make a molecule to actually make it bind more oxygen in the lungs? That actually is not as good of a situation as what the evolution has come up with. What evolution has done instead is it's actually made more of this, it makes more of this molecule that makes it bind less. So if you look at, remember what matters is the difference between the point in the lungs and the point in the tissues. And if you are at, let's say that you're on the blue line, you're at sea level, you have 5 millimolar BPG, and you're at 13 partial pressure oxygen. You're in the lungs and you're right here, 0.95 or whatever it is. When you move down to the tissues, deep down in the tissues, oxygen partial pressures are low, the cells need oxygen, and the binding goes down. You're now at this point, which is 38% below the bigger point up there. This point is, like if this is 0.99, this is 0.99 minus 0.38. Okay, so you can see this is right around 0.6, and the difference between 0.98 and 0.6 is going to be 30.38 exactly. This distance, this 38%, is how much oxygen your hemoglobin is delivering at sea level. But then you move to Denver. You stay on the blue line because you're still making the same amount of BPG, and then you are loading up only at 90%, 0.9. That means when you go off to the tissues, you're dropping to the same level. And that means that you are actually, your hemoglobin is only transporting only 30% of your hemoglobin is being used to transport oxygen. Therefore, you end up feeling out of breath. Your body tells you to breathe faster, breathe more, to try to get more oxygen into your hemoglobin. But what if your body makes more of this molecule, the BPG molecule, that causes the hemoglobin to bind less? Seems weird at first, but actually, if you look at what it does to the graph, it shifts you to the green line. So now, instead of loading up on the blue line here, you're loading up on the green line there. You're loading up at an even lower level, which is probably like 0.85 or something like that. But you're also letting go at a lower level. And because you're letting go on a steeper point in the curve, the difference between the lung binding and the tissue letting go, that difference is back to 0.37 again. That difference is down, let's say is 0.47, and then this would be at, well, I can't do the math in my head, but it would be, the difference between this and this is exactly 0.37 on this y-axis graph. So this is how, when you make more BPG, you do bind less in the lungs, but you bind much less in the tissues. And that means that you're back to using more than a third of your hemoglobin, 0.37 rather than 0.30. And that means that your hemoglobin is good enough to help you catch your breath. So this is as complicated as it gets with this particular chart. Be able to do this kind of calculation. Your book is literally showing you exactly how it works. So look at this, think about this, work on the homework problems with it. Now, in terms of the hill plot, you actually will see the slope of the hill plot. If the slope of this line is changing, the slope of the hill plot is also changing. The slope of the green line is actually shallower, which means the hill plot will also be a smaller slope and a smaller number. And the hill plot is actually going to decrease from around 3 to around 2.5. You are making the hemoglobin less cooperative by sort of forcing it out of the R state and forcing it to have more T state. It's really interesting, and you can think about things in that way. Oxygen transfer has improved. Remember, it's the difference between the y-axis values rather than the absolute y-axis values that really matters here. And we can even come up with a structural reason why BPG will do this. If you have a hemoglobin right here, here BPG is binding in the middle of the T state, and if you look around, you see all that blue. That is charged residues that bind to the phosphates. The P in BPG means phosphate, and phosphates are negatively charged. So you have a lot of positively charged residues that are there in the binding pocket in the T state, and when you look at the R state, that binding pocket has closed up, so the BPG does not bind nearly as well anymore. If you look at the residues, you see exactly what you expect. I could ask you to predict these. There are 3 possibilities. There's the histidine, lysine, and I don't see an arginine here, but it would be the other possibility for putting a residue in that pocket that would have a positive charge to interact with the negative charges of the two phosphates on BPG. And so just remember, if you force the hemoglobin into the R state, that means your BPG binding cavity will be closed and your BPG binding affinity will go down. You'll have less BPG bound. These are opposite effects on each other. So always focus on what are you changing, and then what is that tells you what the cause is, and then follow the chain of reasoning down to where the effect is. So BPG actually plays an important role. If you live in Denver, you make more BPG. If you fly to Denver, it takes you a couple of hours or even days, but you make more BPG. Your body knows how to do this. Now what's really cool is fetal hemoglobin has a very simple change to it. It has fewer positively charged residues in the middle, in the binding pocket. That means at the same BPG concentration, the fetal hemoglobin will have less BPG bound and a higher affinity for oxygen. Think that through. That fits with every step we've talked about. If you have the same BPG concentration, and yet the fetal hemoglobin is going to have the higher affinity, the maternal hemoglobin will give its oxygen, unbind its oxygen, and will allow the fetal hemoglobin to bind its oxygen. This is how oxygen passes across the placenta, because the fetus is expressing a form of hemoglobin that's a different gene with a different BPG binding pocket, less positive charge, higher affinity for oxygen under the same conditions. And this is interesting. I want you to, you can look at this a little bit. You can actually look up this article if you want to know more. But apparently, this is related to why professional cyclists are sort of going through a fad right now, where they're doing the thing that Davy did. They're breathing carbon monoxide. I do not agree with this technique, but they want to inhale a small, precise amount of CO. It will bind the hemoglobin. It will prevent oxygen from reaching the tissues, muscles, and organs. But the idea is that this will sort of imitate the high level, and maybe you'll make more BPG as a result. This will basically put your body in a high altitude mode with less oxygen around. It will basically allow you to prepare for mountain races, and they have one particular mountain race here. But of course, it's going to acclimate the body to using less oxygen. Is this fair or not? Do we want people to be doing this a lot? Should we say this is too dangerous? So whatever, even if it's worth it, that it's not worth it for the long-term harm in health? You can see repeated inhalation of CO can cause all these things that were exactly what Humphrey Davy observed way back. I'm predicting this practice might be banned pretty soon, but it's kind of a weird thing. And like, is BPG banned? I honestly don't know. I don't think BPG injections or eating BPG would get to the right place as easily. The thing about CO is it goes through the lungs exactly where it needs to go. Anyways, bad idea, but you can see their reasoning. Just because something has good, reasonable reasoning behind it doesn't mean it's a good thing. Okay, so a shorter story in this case, but there's a story about Methemoglobinemia, which is an application of what can go wrong with hemoglobin biochemistry. And this has a striking difference. You know that veins and arteries have different colors. I believe part of that is that iron is going to be in a slightly different oxidation state. Remember how we're keeping it in the iron 2 oxidation state. But it can flip over to iron 3, and when you get into iron 3, it's hard to get it out of the iron 3 mode. And it also turns to be more bluish. You see how it's more purplish in this particular thing right here. What do you do if a patient has too much methemoglobin, iron in the +3 condition? Well, your body normally will use cytochrome and it will use NAD to be able to convert it back from methemoglobin to hemoglobin. So we can help it along by doing something else. It turns out that methylene blue is a dye. It's a blue dye. That's just a coincidence. But it's a blue dye that can actually accept the electrons from +3 iron. If you give this blue dye to someone, their iron will convert from +3 to +2. It will actually donate electrons to it. I think I might have said it backwards. It will give it electrons so that the electrons will turn it more back into iron too. Methylene blue is strong enough at donating electrons as able to do that. And it actually interacts with the body in NAD as well. This is the full reason why if someone has too much methemoglobin, you actually give them methylene blue to change it. And actually, when you have too much methemoglobin, sometimes in extreme cases you turn blue. And you can be fairly healthy in other ways. You can go on talk shows like this guy right here. This is not a touched up photo, and this is also not a touched up photo. There's a bunch of people who had similar genes in Kentucky that had a methemoglobin problem, and they would be blue, especially I believe when they got older. Although it can show up in the babies. If you see a baby that is too blue, they might have a problem with methemoglobin. So what do? You give them the methylene blue so that they can convert their iron 3 back to iron 2. There's a summary of this. There's effects and causes of methemoglobinemia, but they actually show the wrong direction of conversion because they're showing NADH being converted to NAD, which means it's giving off electrons. The electrons will actually go the other way. So this right here is reversed because you should be giving electrons to the iron 3 and turning it to iron 2. But if you look over here, you do see an example of our favorite curve that we're talking about, partial pressure of oxygen, oxygen saturation of hemoglobin, and you have methemoglobinemia is going to have a higher affinity for oxygen because iron 3 has such a high affinity, it's actually irreversible. So the more of those molecules you have in your blood, the higher your curve is going to be. And it is coincidence that the safest way to supply electrons is methylene blue. It's self-blue, so you cure blue people by giving them something blue. I'm sure the first person to try this had to convince a lot of people this was the right thing to do, but it's one of the oldest organic dyes. Scientists just made this structure. They saw it was blue. It gives off electrons really easily. It's also very safe for the body. And so, if you want to give the iron more electrons, you prescribe this methylene blue. And you give someone a blue drug so their skin turns away from blue. Now there's the final one, which actually composes both of these, the sickle-cell anemia, which is probably the most important. And this is also geographically distributed, so it does have a collection of genes that shows up in a certain subset of people. It's important for doctors to know about this so you can see past your own inability to experience this, depending on which side of the genetic distribution you're on here. There is a form of a gene of hemoglobin that is predominant in African people that will cause sickle cell anemia. So the thing to realize about red blood cells, they're very flexible. So for example, like camel red blood cells, they look very different from human ones. They're smaller, they have more protein, and they're more rigid. The thing is, the camel has special membranes, and they are able to actually absorb so much water. Camels have such huge swings in the volume of water that they have inside them, that their blood itself has evolved to be more rigid and to allow it to swell more when the camel drinks 50 liters at a time. These will flow differently. Camel blood will actually flow like water rather than human blood, which is much thicker. So it's interesting. Blood can be very different. It's the same hemoglobin doing the same things, but the other parts of the blood can be very different. And so when you have changes in the viscosity of blood, you have health problems, especially if you have not evolved to adapt to those. And for example, there was a dental student from Grenada who was presenting with anemia. And so they knew that this student had anemia, went to the doctor at the dental school, and the Chicago doctor looked at his blood and he found out, oh, there's all these things that shouldn't be there. But these are not different organisms, these are the patient's own blood cells. They look like sickles, and so said it was a form of anemia called sickle-cell anemia. Turns out the patient doesn't have an infection, the patient has a genetic mutation in their hemoglobin that makes their red blood cells much less flexible, more like camel cells, but they can't deal with the rigid, sharp cells that result. So they looked more, they found the genes for it, and they found out that actually a lot of people coming from East Africa, Central Africa in particular, not the extreme south or extreme north. And so this tells us there might be something else going on here. So somebody who has is homozygous for a particular mutant will develop sickle-cell anemia without a doubt. Somebody heterozygous, if they have one unmutated pair without the sickle cell mutation, they actually won't have sickle cell because it's like recessive in that sense. And they also will be protected from malaria. And it turns out that 1 in 12 Africans are heterozygous. Well, if you do the math, if 1 in 12 are heterozygous, that means 1 in 144 are going to be homozygous. And that is the case. About 1 in 100 to 200 people will have sickle cell anemia in Africa. And that was something that the doctors that I know in Africa, you need to know how to deal with this. So sickle cell is a disease that affects the African American population more. If you're an American doctor, you need to know about it. And we can do sequencing because we can find the exact mutations that do it. There's one simple major mutation we'll talk about. There are actually, if you look at hemoglobin genes, there are 300 major variants, and these are called alleles. So remember that you have 2 alleles of every gene, except if they're on the Y chromosome. These aren't. And if you have 2 of the same, you're homozygous for it, 2 different ones heterozygous. And sickle cell anemia develops when you have 2 of the same mutation that causes a problem with the hemoglobin. So here's the anemia signs. Physical exertion is weakness, dizziness, and shortness of breath. Hemoglobin content is half normal because the sickle-shaped cells rupture easily and they lose their hemoglobin. Also, the sickle-shaped cells are going to block capillaries, and this is where pain and even loss of organ function comes from. Think about it a little bit. What is the organ that has small capillaries that the blood passes through in order to be filtered? It's kidneys. So you would predict from this that sickle cell anemia would have problems with the kidneys, and that is exactly true. But it's better to look at it, and here's 2 different ages of sickle cell anemia sufferers. And it's by all means, it talks about a lot of pain, unbelievable pain, like a jackhammer on your back throughout your whole body. Because what's happening is there's an episode where you have a bunch of sickle cells and they're getting caught in all your capillaries throughout the whole body. But the doctor can't see that. So you come in and you tell the doctor that you're in pain, and they say you're a drug seeker because they can't see the pain, right? So it's important as doctors to realize that there are some very significant syndromes where you can have this kind of pain like this, and it can be real. And you're not, the people aren't drug seekers. They are suffering from sickle cell anemia. Here's an 8-year-old, and basically the episodes of sickle cell actually coincide with fever, like the immune system starts to freak out because the whole body freaks out and you end up with a fever. And so the 8-year-old says that when an episode starts, her head starts getting warm, her eyes sort of turn yellow, there's some kind of jaundice to the eyeballs, pressure on the legs and arms, and it can be very, very painful. doctors need to know about this so you can look for it. Because you don't have time to do genetic sequencing when somebody's in the middle of one of these episodes. So we call the mutated hemoglobin hemoglobin S. And it's S for sickle cells. We'll compare it to hemoglobin A as the most prevalent non-sickle cell hemoglobin. In hemoglobin S, there is one consistent change. A glutamate has been changed to valine at position 6 of the beta chain. So we can represent those with 4 characters. Beta E6V. That one little change actually can cause the entire disease. The reason is because hemoglobin S has actually a different tertiary structure, because that little mutation has caused its helices to sort of space out a little bit more. It's got the same helices, same spaces, it's got a different primary structure, and its tertiary structure has changed just enough so instead of being shaped like a dome, it has a hydrophobic sticky patch on the beta chain. If you have that sticky patch on the beta 1 chain, you're also going to have it on the beta 2 chain, which means that you don't just have one sticky patch, you've got 2. And that means when they start to run into each other, and often dehydration will bring on a sickle cell sort of process, once they start to run into each other, they will end up forming a fibril, not of beta structure, but of hemoglobin molecules. So the sticky hydrophobic surface shows up on both sides. The molecules stick together. They form a strand. Enough strands form. They start to wrap around each other. They start to crystallize into a fiber. And that gets to be big enough that you can see it. That gets to be big enough that this thick fiber is forming inside the flexible red blood cell. And it starts to basically poke out of it. And that's where you get the sickle cell shape from. You can see there's a sickled cell on the left right here, but also you can get all sorts of pokey little shapes. They're more fragile and they break more easily, so you might not even see all the shapes. You just know that there's fewer red blood cells because some have broken. Capillaries can sometimes be so narrow that just one red blood cell can barely fit through them, like it has to be flexible and squeeze through it. It's like spelunking in a cave. If you have the sickle cell making it wider and more fragile, it's going to try to squeeze through. It's not going to fit. It's going to block it. It's going to break. And then you have the pain symptoms. And this is why, because the capillaries are the site where the most damage happens, this is why kidneys are commonly dysfunctional in sickle cell disease patients. And you got to realize there are other things as well. There's lots of things that can go wrong. You can end up with gallstones. You can end up with problems with your bones. The biggest thing is this chronic pain that is systemic, but you can't locate the cause because the cause is in the red blood cells. It's at the molecular level, which you cannot see. So there's ways, how can we detect when we have more sickle cells, short of taking a blood sample and running over to the microscope? Well, there's some people that are even suggesting the red blood cells with sickle cells are so different that they even sound different. If you like ping them with sound waves, you'll hear different echoes back because the flexible ones are like a drum and the non-flexible ones are like a tin pan. I think this is brilliant if it works. I'm not sure it works, but it's based on a cool idea. But a simpler diagnostic, one that I can actually envision using in Burundi, would be something where you take a bunch of tubes and you can get a centrifuge. You can do one even without electricity. If you can get something in a tube that would work with the centrifuge, you can use density to separate out the cells. And so here is an idea that people have had. People have tried to develop these things. This is a tube with just a thick gel in it. And the gel is thick enough to separate out normal cells, flexible ones, from the rigid sickle cells. And what you see is like you add blood to the top of the tube, then you centrifuge it to spin it down, and the blood gets separated, the red blood cells get separated out, and you can have red blood cells in the middle that are flexible, and the ones that are really dense and inflexible can be pulled all the way to the bottom. I believe those are the sickle cells. Yeah, so for example, here you have a tube of homozygous hemoglobin A person homozygous or heterozygous AS, and then you have the homozygous sickle cell person, and you see right down here in the dense part, you see a bunch of red cells. Hopefully you can see that. I'm actually going to move it over so that I can make sure that you can see it on this screen. There it is right there. These are the dense sickle cells, and it's based on just the fact that sickle cells are more, they're misshapen, they're dense, and they don't work the way they should. So just remember that these things, if you're a doctor, you're looking for the presentation of these things. There are other kinds of anemia that might be caused by something else. But all these things are caused if you have a hemoglobin S patient. All this is caused from a single point mutation, which is amazing that a small change like that can cause a big change like this, but it also gives a certain kind of hope, because it means if there's just a small thing that's wrong, maybe a small drug can fix it. So there's a lot of sickle cell associations. We are working on the disease, and I'm going to tell you some ways in which we may be making progress. But there's also, like, SPU Stories in 2022 actually interviewed a sickle cell patient and talked about how SPU is giving them voice. And I hope that as part of the medical establishment that you can go and maybe you can work on either treating patients like this or maybe even coming up with a cure for sickle cell. Here are some things people are trying right now. One of the ideas is that a small molecule might be able to block the sticky patch. So a drug company has come up with this voxelotor molecule that, for whatever reason, they claim will stick into the patch. And so they've shown that it sticks into the right place. They've shown that if you have a lot of it around, it will prevent the fibrils forming. And I think that's very cool, but I'm not sure that you can get it in the high enough concentration to actually do much good to a patient who's in the middle of this. Now, that's because it's not directly blocking polymerization. Notice how I said that, and I assumed that I remembered how it worked. I was actually wrong. It does not bind at the sticky patch. That's not practical. If you think about the problem, the drug shouldn't work that way. The problem is that hemoglobin's concentration is 3 millimolar. Half that would mean that for a perfectly binding drug, you would still have to add 1.5 millimolar of that to reach 50% of the hemoglobin bound. So you'd need a drug in the blood, assuming that it all gets past the membrane. That would mean a drug concentration of 0.45 grams per liter, 450 milligrams per liter of blood. 5 liters of blood, that means a single dose would be 2.5 grams. That is too much. We need milligram doses of drugs for them to work, not gram doses. So my memory was wrong. It's not directly blocking. Let me see. It actually will increase hemoglobin's affinity for oxygen, which will promote the state that is less sickle cell forming. That said, I'm not sure about this. I hope it works, but I would put this low on my list just because of these kind of problems. One other thing you might think about, what about gene therapy? This is a targeted problem. Maybe we can replace this gene, the sickle cell gene, with a gene without the mutation. And in fact, sometimes you don't do direct replacement. Sometimes you replace another gene. There's various reasons why you would do this. Here's an example where there's a one-time gene therapy that will give you a beta globin gene containing T87Q that will actually prevent fibril formation. That way, you can even be like heterozygous for this, and it will prevent the sticky patches from continuing. Maybe a few of them will form, but then statistically speaking, you will run into this T87Q, and it will make the sticky patch less sticky. And it looks like this is working. They're giving it, and you see that there's actually a good amount of the red T87Q around it, and it's half of the genes. The other half are the HBS gene. But if you give a patient this, they actually still have the gene, uh, 36 months later, and their total hemoglobin is still high later. This might actually work. This is 24 months before enrollment. Each of these is an individual patient. Each little red diamond is a sickle cell event. After the lentoglobin infusion, the diamonds go away. They had 0. Everyone who received the good mutated hemoglobin A actually continued to have 0 events after the fact. That's pretty good looking. And so I need to follow up on this to see if the good news is still there. But one of the questions is, why is it focused in Africa? If you look at just overall births with sickle cell anemia per 100,000 people, and there's around 1,000 per 100,000. If you look, that's about 1 in 100. That's about the statistics that goes with the numbers that I was quoting earlier. In the central region of Africa, which includes Burundi, where I taught for a short while in 2015. So I knew people, and probably some of my students at the time had this mutation. In fact, I'm sure some of them had it. The only thing I'm not sure is if any of them were homozygous for hemoglobin S. So here's another statistic. This one says 8%. That goes with the 1 in 12 I quoted earlier. And they've always known that hemoglobin S and sickle cell anemia tend to show up also where malaria is endemic. And so this is a hypothesis. And it was a question mark for a long time. When I first started teaching, I had to put a question mark. Well, guess what? I can take away the question mark because we now have a bunch of studies showing hemoglobin S heterozygous will actually protect you against malaria. It's why the gene persists. Here's the paper. If you want to go into it and you want to find out exactly why, here's the paper that really is the central paper to this. Sickle cell hemoglobin confers tolerance to Plasmodium infection. And they did some molecular biology to figure out how it confers tolerance. It looks like the fibers, when they form in a moderate level, they don't bother the patient as much, but they really bother the malaria, which is trying to live and do its life cycle within the red blood cells. So it looks like particularly the little halfway fibers that happen will interfere with actin remodeling, and it means that the Plasmodium, the malaria-causing organism, will not be able to reproduce nearly as well. If it doesn't reproduce, the person doesn't suffer from malaria to the same degree. They even looked at what exactly in the red blood cells. They have a tension. This is not surface tension. This is literally the tension of the red blood cells. Because they are more tense in general, it actually prevents the parasite from invading them in the first place. If this is held up, as this is held up, we can try to recreate this with a treatment that does not have the side effect of the occasional bouts of sickle cell disease. And in fact, malaria is still endemic, evolution still works, and sickle cell is still being selected for in Central Africa. This is just this one particular gene. This is a very focused case, very narrow case in which evolution is still happening. And it's basically the arms race. It's the same kind of thing that goes on in everyone's immune system. You are constantly evolving because you're fighting things that are themselves constantly evolving. So this is what the clinics look like. If you ever do global medicine, you can see that they actually will advertise what they do on the outside. And if you look up and down on this, you can actually find biochemie, hematology is here, and I believe sickle cell will show up somewhere on some of these lists. This is in the Democratic Republic of Congo, and it looks just like the hospitals and clinics that I went by in Burundi. And there's lots of stories about this. Gene therapy is great if it works, but it's hard to deliver in Africa. Can we come up with a better kind of treatment that can treat people who carry the HBS gene? And there are certain families that, for example, this family has lost 3 daughters to sickle cell, and there's a 4th that is suffering from it. So the question is, what kind of therapies can we develop for people in this situation? Not gene therapy, but something simpler. That's why that small molecule is worth looking into, even if it's got an uphill battle. Again, this is what it looks like. This is the kind of place where the medicine is practiced, and we need medical treatments that will work here, as well as medical treatments that will work in the snazzy, expensive American hospitals. But that's one thing science can possibly do. If we can make a diagnostic that works, maybe we can make one of these that works, a treatment that works. So and in fact, it's not just an African problem, because hospitals with fewer resources in the U.S. also will have trouble providing the basic care. And so a better gene therapy is also out of reach for many people in the U.S. One thing to do, there's many ways to work on that. One way to work on that is to come up with a better, cheaper treatment. So if you are interested in that, I encourage you to look up those articles, think about this, and think about the big picture, especially if you're going to be interviewing about this kind of thing someday. But I do want to say that actually one of the promising treatments of sickle cell involves something already mentioned, fetal hemoglobin. In fact, they've known for a long time, and I think this is just by chance, they've known for a long time that this molecule can induce fetal hemoglobin and can reduce the fibril formation. Hydroxyurea will stimulate the production of fetal hemoglobin. It actually does a number of things, so it doesn't do just that. It'll also disrupt the fibrils directly. But you will see higher levels of fetal hemoglobin when you're messing with hemoglobin in general. Sometimes your body will go and turn on the fetal hemoglobin gene because it says, I need more hemoglobin. Well, what's really cool is everyone has a separate HbF gene, right? So if you have a mutated HbS gene, and you have two of those, you actually still have a fetal hemoglobin which is not mutated in the G to V mutation, E to V mutation. So if you can just turn that on, that would get in the way of fibril formation because it does not have a sticky patch. So gene editing could work. How do you do this? Which gene do you go after? One of the ways to do it is to insert the gene into bone marrow. If you do it into blood, it will just be replaced, and blood doesn't even have the machinery to do this. But if you do it into bone marrow, the cells that make the blood, you can actually try to turn up their HbF levels, and if you can do that, you can actually have some approach toward this. There's also CRISPR to edit whole genes. Again, that's more molecular biology, but we have to mention that a lot of people are doing this, and some of this is working. And so some people will actually have a deaminase that will edit not the HbF gene directly, but the regulatory DNA that switches the HbF gene on and off. Anyways, it's really complicated. If you want to get into it, I want to give you the resources for that, but this is more molecular biology. We want to talk about the biochemistry more. And we want to think about, could there be a pill that could do this? So when you're born, the fetal hemoglobin gene is turned off by the binding of this one particular transcription factor called WIS. So, and in fact, it's kept off. Right now, you have the WIS transcription factor that is sitting on your fetal hemoglobin gene and keeping it down, keeping it off. What if there's a small molecule that can sort of tag WIS for degradation, so the cell goes through and says, chops it up, opens up the fetal hemoglobin gene, and then all of a sudden you have all this fetal hemoglobin. And it's possible there's a lab that says they've developed a drug that does exactly this. The really cool thing, it's not gene therapy, it's not a protein even. It's a small molecule pill that you could take once a day. So let's realize that sickle cell anemia with CRISPR, you can look into that. We don't focus on CRISPR, but you can even talk about possibly editing the mutation, the single mutation. It's actually a single nucleotide that changes a single codon that changes a single amino acid. But you have the beta globin gene here. You can replace a T with an A, and you can do that with CRISPR, and those could make normal red blood cells. But rather than fixing the broken gene, maybe you can add an unbroken gene that will just tilt the scales and make the disease not happen. And that's the idea with fetal hemoglobin, turning that on, removing what's turning it off. Details are molecular biology, but I want to say there are these papers if people want to do this. This is the idea of some people who think they have a good approach for CRISPR to disrupt the gene signaling and to turn on more fetal hemoglobin. But again, more genetics. I do want to give you the ability to look into this, and it turns out there's even a whole webinar about this. So if you're interested in how biochemistry and sickle cell go together, watch this webinar. I haven't had time to watch it yet myself, but it's from a very trusted source, and I would like to know what you find out about it. This would be a really good topic for another class. This class we're going to focus on, we've got enough for test 2 right now. And there are particular scientists, even this scientist from Liberia has a calling to use chemistry to try to come up with small drugs that will work against sickle cell. This scientist was profiled, and her current goal is to increase fetal hemoglobin. Currently, normal fetal hemoglobin is less than 1%. If we just increase it to 15%, that's well within range of what a drug can do, and that would reduce the severity of the sickle cell episodes. And in fact, some people think that we can get up to 30%, which would really reduce the severity. So the gene therapy is a problem, but she's working on this for particular. She's targeting that same transcription factor, and the idea is finding ligands that will bind that transcription factor. Once you've done that, that's biochemistry, once you focus in on that part. And so there's, this is pretty old. There's been a couple of years. There's been a bunch of progress with sickle cell, but I haven't heard as much about it lately. I'm hoping that the progress is still there. You notice that there's this New York Times article. This was back in the Biden administration, so several years old now. And the question is, where is the field now? And I know that some of the researchers have had their funding cut, and that's why I want to say to you, well, if you want to do something about these things, you can work on the funding end of things, you can work on the political end of things, or you can work on the scientific end of things, where you can try to develop a small molecule on your own. And we can do projects here at SPU sometimes when we find the right project, where we can actually contribute to these things. So you're learning about this in the lab. I want to say that this has been a bit of a long lecture, but I want to show you how this is worth it and how this can help people like the sickle cell patient in front of you. If we can do something to help her, then you've really made a difference in this world, and you've really alleviated suffering in a way. In the meantime, if you don't want to contribute in the lab, you can contribute on the streets or in the houses of the people. There's a whole Metropolitan Seattle Sickle Cell Task Force. If you're looking for a place to volunteer for pre-med type stuff, look into them specifically. We've had students who have worked with them before. You can volunteer with those patients and find out what life is like for them and help alleviate their suffering in some small way along the way. So thank you for that. Hopefully that's a little bit of a call for action. But again, this is 1 hour just to finish off test 2. Wednesday, we're going to talk about the reading. We're going to talk about the rest of the test. We're going to review for the test. Friday is the day. So hopefully you are watching this and thinking about this. And I'll ask you some questions about it in a couple of days. So I'll see you then.